Hey everyone, and a huge welcome to our FUTA undergraduate agriculture students. You're tuning into the explainer for Practical 1 of CSP 204, Botany of Economic Crops. On behalf of your incredible instructor, Dr. Lawrence Stephen Fayon, I am so thrilled to guide you through this material today. Let's jump right in. Today, we're laying down the absolute essentials you're going to need before you step foot in the laboratory. We'll quickly cover basic plant cell structure, walk through exactly how to prep an onion epidermis slide, and then we'll compare how tissues are arranged in monocot and dicot stems and roots. All right, every single plant you'll study is built from cells just like this one. Look at the diagram on your screen. You've got that rigid cell wall giving it shape and support, while the inner cell membrane is kind of like a bouncer, controlling what gets in and out. The cytoplasm is where all the cellular activity happens, and the nucleus is your control center. And notice that massive central vacuole right there? It basically stores water to help the whole cell stay nice and firm.
Now, when you actually prepare your onion slide in the lab, it's a simple three-step process. First, just pop a drop of distilled water on a clean glass slide. Next, grab your forceps and carefully peel a super thin, translucent layer right from the onion's inner surface. It literally has to be thin to prevent the cells from overlapping. Place it in the water and then slowly lower your cover slip at an angle to avoid trapping any annoying air bubbles. Notice that we aren't using any stain here. We want to view these cells exactly as they naturally appear.
Okay, moving on to tissue-level organization. I actually want you to pause this explainer right here. Compare the two stems on your screen. Which one shows vascular bundles arranged in a distinct ring? Take a second to decide. All right, ready? Over on the left, the monocot stem has its vascular bundles just scattered all throughout the ground tissue. But look at the right. The dicot stem has vascular bundles forming this beautiful, distinct ring, completely separating an outer cortex from a central pith. Honestly, looking for this ring is the absolute fastest way to tell them apart under your microscope.
Let's observe the cross-section of that monocot stem in a bit more detail. Notice how scattered those vascular bundles are? Each one contains both xylem and phloem, all wrapped up in ground tissue, with the epidermis as the protective outer boundary. When you're viewing your own slide in the lab, see if you can actually count the individual vascular bundles. It's a great way to train your eye.
Now look at the dicot stem. Notice how those vascular bundles perfectly form a ring? The phloem sits toward the outside of each bundle, while the xylem is packed toward the inside. This ring just beautifully separates the cortex from the central pith. When you compare this highly organized setup to that scattered monocot stem we just looked at, the difference is literally night and day.
So let's head underground to the roots and see if those stem patterns hold true. Time to pause the explainer one more time. Predict for me, which root possesses a distinct central pith? All right, have your answer? So both roots actually share the exact same basic sequence from the outside in: epidermis, cortex, endodermis, and then vascular tissue. But look closely at the core structure. The monocot root on the left actually has a distinct central pith. Meanwhile, the dicot root on the right has no distinct pith at all. Its vascular tissue takes up the very center instead.
Now, the endodermis is one specific layer that is common to both of these roots. Direct your eyes to this single ring of cells. It's an absolute game changer because it controls water and mineral movement right into the vascular tissue. You see, water can't just flow freely between these cells because a specialized band called the Casparian strip completely blocks that route. This essentially forces the water and minerals to pass directly through the cell membranes, acting as a strict filter for whatever reaches the plant's vascular system.
All right, remember these structural differences, and a quick pro tip: always start your microscope observations at the lowest power. Now, before you head off to the laboratory, look at the Think and Reflect panel on screen. Ask yourself, why are chloroplasts absent in onion epidermal cells? And how does vascular bundle distribution differ between a monocot stem and a monocot root? Think deeply about these two questions. What do these tiny differences actually tell us about how these plants survive? Come prepared to discuss your answers in the practical. See you in the lab.